Excellent. So, thank you everybody for joining us in the room today. And I know many people will also be watching this on YouTube later. Just wanted to give a quick introduction here. So, this is what we've kind of been calling HUBscope Chat or HUBscope Session number one. Today, Tanja and I have both put a few slides together, and then we'd like to discuss the concepts with everyone in the room afterwards. And then next week, I've actually already put on the Holobiont Hub calendar Hub track number 1 and 2, which will be chances for me to get feedback on some presentations I'm working on, one on grasslands and one on nutrients in soil. But Tanja, I'm going to switch this over to our opening sequence here and then let you take it away, and you can let me know if I'm not keeping up with your slides.
Good. Yeah, I want to talk to you guys about thinking like a soil detective because I think microscopy is so much more fun if we had that attitude. So let's go and decode soil health with microscopy. So, you guys, why are bacteria so bad at working out math problems? Well, they have to divide to multiply. It's a little comic relief we need every once in a while. So, what we need to do if we do microscopy is we need to move beyond counting microbes. The microscope is a tool, but it does not work in isolation. A soil detective integrates knowledge and surveillance, and we're talking about soil structure and organic matter and microbial interactions and ecological succession in this sense. So let's combine scientific analysis with careful observations, because that's a way to uncover patterns that reveal soil function and soil health and the needs the soil actually reveals to us. So this requires a shifting between reductionist, a quantitative, and a holistic, qualitative thinking. So concepts. Concepts I want you to have in your mind when you are doing microscopy. So soil is a living and a dynamic system, right? Microbes don't exist in isolation. They function within a larger ecosystem. And soil is constantly changing because it's shaped by succession and the organic matter inputs that we put in and the disturbances that we cause. We have a soil food web, and the soil food web has ecological functions and microorganisms, and we need to look at them and see who dominates and what do they tell us. So let's look at the trends. The other concepts I think you should hold in your minds is the soil structures. Of course, it does say something about your F to B ratio, right, across succession, but it's the soil layers that are very important. We need to pay attention to our O horizon and our A horizon because that's where the roots are. And then below that, we have a mineral zone, which is also very important, but it's not a good thing, as we have noticed, to have things grow straight in the mineral zone. We need to look at the O and A horizon and help it recreate. So who is running our below ground civilization? What are we looking at? Every sample tells a story, and we need to uncover that story. We can connect the dots because what we see is what we can connect to what the microorganisms indicate. And we can solve the case, and that's how we can restore the ecosystem. And that's what the thing is. We need to go from microscopy ultimately to land management, which looks like a very large leap, but it's something we can definitely do. So I love for all of us to become soil detectives, and we need to link our observations to our interpretations. And a detective doesn't just see the microorganisms, it's connected to the other things that we know about it. The real skill is connecting assessments to functions and then connect them again to our management strategies. And every sample tells a unique story, and I'd love to uncover it with all of us. So let's take it further. I'm inviting everyone to participate in this microscopy and soil structure. I want it to be fun, but I also want it to be relevant because, I like relevance. So what I want to nerd out on with you guys is I want to go through these soil science concepts that we need to keep in our minds. We need to have a framework that we agree upon, and then we have a language, right? Most of us have this language. I'd like us to bring our microscopes and do life assessments. What are we seeing? What are we noticing? I want us to interpret biological communities and link it to real situations. And I want to connect microscopy results to soil health and, of course, what to do in our land management. So I'm inviting all of us to become true detectives. So what do you say, you guys?
Awesome. Thank you, Tanya. And I have to say that, you're so much better than I am at thinking through how to get new people excited about this as well. So I'm really glad that, we put your slides in the front of this, because it's, the YouTube video will be kind of an announcement that this is going to be happening over time and that we're holding these free sessions. And I want to dive into some really technical stuff because, I put out that hub note a couple weeks ago on the microscopy and some of the pitfalls that I saw, and yet I was trying to reach for something where we could tap into the collective intelligence of this group, right? So let me just quickly run through a couple of ideas, and then again, we'll stop the recording and go back and have a bigger discussion with the group. So, um, I mentioned the idea of an index of biological integrity and how there was something already like that for wetland ecologists to use. And so I want us to think, as we go down the rabbit hole together, what comes next? If I say the current thing has some flaws in it, the quantitative mindset takes us down some pathways that are problematic. I'm going to ask this group to go through with me and say, what would a soil index of biological integrity, or a SIBI, look like? And I really want everyone's perspective. This uh, like piece of artwork here on the side about the soil food web. We actually made that when I was at the school, and I was tapping into 6, 7 people's ideas constantly going through, like, have we thought of everything? Should we, well, technically a nematode is a kind of microfauna. Should we collapse them into one category or should we separate them? And I'm pretty happy with where we ended up, just as an overview of the soil food web. So I want to do something similar for microscopy. And that really is a matter of saying what are the targets going to look like, and if we aren't so, I hope we're going to get to a place where we're not so focused on measurements, which can be quite biased, but that we have a really good overview. So diving into just a few slides here, a few concepts. Hope everybody will take time to read this article that I found, open access online. I've put the address down on the bottom there because I think the starting point for a lot of the microscopy discussion out there is sort of like, oh, we have to identify who's good and who's bad. I really don't think that that is going to ultimately be the mindset that we need to be at. There's a lot of uh, research going on in the health field right now about the human gut microbiome, and it's not all about who's a good guy and who's a bad guy in there. And I think that we need to recognize that there are things like induced resistance in plant hosts, where the presence of a small amount of something that could be pathogenic can actually make the plant stronger, right? And so that's just a little plug for their work here. So I'm going to go through a couple of categories here of things that we would be able to see through a microscope, and starting with bacteria. And I found this very zoomed up um, picture of, I believe, Listeria. And I don't think that through a 400x, um, total magnification, I would trust myself to not just identify that as a rod-shaped bacteria and maybe say it's totally fine. It looks great. And so there's things about the complexity of the morphology itself that we see. I'm going to highlight a couple of things that I've been thinking about over the last few years. I think actinobacteria sometimes get a bad rap. I've actually heard people say that like they're antagonistic to AM fungi, so we don't want them, quote unquote. But I've found papers that say that they can also be complementary, cooperative organisms with AM fungi. And I really wonder if there's just kind of a crossing point in succession where actinobacteria become relatively less abundant and AM fungi become relatively more abundant. And the interpretation was, therefore, we don't want actinobacteria. But again, going back to a perspective that everything can have its place, right? And then the concept of morphological variation, which I've actually put in the formula for Shannon Diversity Index. So people often say, we want diversity, which is quite a bit more complex than just saying, oh, we have this variation in the shapes of the bacteria. But I think it's really important that we keep in mind that species ID is too complex to see through the morphology of bacteria, especially. Um, Experts might be able to tell you exactly what species of nematodes you're looking at because they've trained for years to notice all the features. But I think variation in the shapes is going to be like the closest metric we could get to. And then, there's certain shapes that I don't like to see in my compost, spirilla, spirochetes, vibrio. And actually, I found that all of them, I had this concept that they were all anaerobic organisms. And in reality, they're quite facultative or microaerophilic. They like reduced oxygen, but not necessarily like 1 part per million or 3 parts per million, like we'd think of for the true anaerobes. And so I just want to have a discussion with this group about the realities of bacteria. Um, Likewise with fungi, I have some real issues with so the general trend that fungi would increase in abundance and bacteria would be a little less dominant over the successional um, um, transformations in an ecosystem. It's generally true, generally a true thing or like an observable trend. But I do not like counting bacteria. I think a lot of times there's a lot of bias in being overly focused on getting the biovolume exactly right and micromanaging that. And in fact, I found this paper where they took one isolate, like one species of fungi, and they grew it in this interesting foam so they could get all these 3D like cuts of it. And actually, some of the things that I have always thought about regarding, oh, larger diameter fungi are, more beneficial to plants or whatever. It can also be the age of the fungi that they can appear quite small. Some of these hyphae came out at 2 to 3 micrometers in diameter, but the same exact species had a lot of examples of very very thin diameter. Um, and then spores, sometimes these things like I've put an example here of AM fungal spores. They're quite variable, and I want to see that variety. And if we take just this idea that oh, it's a spore of a certain size or a certain color, it's therefore mycorrhizal. I don't think that that's necessarily going to be as informative of the reality as seeing this like general kind of variation again in the spores. Um, I want to point out too, omycetes get lumped in with fungi a lot, and they are in fact protists. They're more related to algae than fungi. They look a lot like fungi, which is why I think people make the confusion. Um, so yeah, what would we use fungi as meaningful indicators? Like I want to see abundance and variation in color and shape and size. And I do know that most filamentous fungi are aerobic. And so if I see this huge diversity and array of fungi in my worm compost, I think I probably got the oxygen diffusion, principles kind of right here. And there's likely to be the kinds of other organisms I want to see. Protozoa are interesting. I hear people act like ciliates are bad guys again, and I think that's very interesting because there's a range, or they're very adaptable to different oxygen preferences. And in fact, this paper I mentioned, they say that the flagship soil ciliate is actually quite an aerobic organism, that it likes that it prefers these high oxygen or higher oxygen environments. And this older paper actually came up with like less than 2% of soil ciliates are going to be true anaerobes. Um, so there's a huge variation in ciliates. I don't even know that I would trust myself to pick out that each one of these in the picture is a ciliate. There's other things that fit under protozoa, right? Of course, amoebae, testate and naked amoebae. Um, they are good indicators that somehow you have your trophic ecology right? Because they're predators. So even at relatively low numbers, they confer a really important ecosystem service in grazing on the bacteria. Some of them can eat fungi. They can sometimes hunt each other or nematodes as well. So again, I would focus on, if you see this diversity and array of protozoa, are you going to have, is that going to indicate to you that you have an environment that's, got a kind of a functional soil food web and aerobic conditions? And then microfauna are really interesting because I've seen rotifers, tardigrades, lots of nematodes in my worm compost. Rotifers can be tolerant of very low oxygen. So if I saw them and nothing else, I might say, "Ooh, we might have an issue here with like a kind of hypoxic environment." But nematodes can be variable from down to almost like below 0%, or like just a few parts per million oxygen, all the way up to 14%, right? If the atmosphere is 21, then there's a diffusion into soil. And even though I like, in general, some of the categories that people talk about, I think that this makes sense to try to put nematodes into these general categories of trophic, what they eat. This paper from 22, uh, it blew my mind because it had so many examples of things that I would have miscategorized that. I would have thought that was a predator. I would have thought that was a fungal feeder, and it's actually belongs to a different category. So again, if we put the stress on the measurement, then we have to get absolutely every single nematode ID correct, or we might, come to the wrong conclusions. But if we're looking more for diversity, and if we accept that some of these organisms take years of study to learn how to identify, then they can still tell us something about our soil, right? If we go from no nematodes to a bunch of different nematodes, we're going in the right direction, right? So, of course, as multicellular organisms, tardigrades, nematodes, they tend to need some oxygen because of their respiration. And so, you know, again, I'm looking for them in there. I mean, when I put my worm compost for the first time on a slide and saw it crawling with a bunch of different tardigrades, I got really excited because A, they're very cute, but B, they're a highly adaptable organism that I expect, if you give space for it, will be able to fill a lot of different microbial niches or microorganism niches. Okay, I'm almost done. I know this is running the risk of throwing a lot at you, again, we can stop and discuss each of these things step by step. But if we're talking about what a key performance indicator for soil regeneration would be, we have to keep in mind that a lot of agriculture is just going to be in one of these sort of mid-successional phases. I mean, there are bushes like cotton is an example, or um, blueberries. There are uh, trees, there apple trees and things that are going to be in later successional systems, more fungal dominated. But a lot of what we grow, a lot of what I grow in my garden, a lot of field crops are going to be in that sort of mid-successional um, gradient. And that's actually a good thing because this paper about succession that goes all the way back to the 70s started to say like, well, if one of your goals is species diversity or maximizing that, then it actually tends to peak in these systems that are quite diverse and mixed in the middle of succession. And as you go along the gradient and get into the dominance of just a few kinds of trees and a few kinds of fungi, then you're actually going to see the diversity of your microbial community, your insects, a number of different things decline, right? So I want us to have that discussion about succession, but also understanding this dynamic. And I put out there that we may want to have some kind of a scale that helps us, say, oh, if you're between, move this slider. Are you seeing two shapes of bacteria? Are you seeing 12 shapes of bacteria, right? What would that look like as a score? I think we could actually get away from numbers in general and look at something from more of the social sciences world, where our descriptions are more like the diversity is minimal, the diversity is moderate, the diversity is substantial, right? And we kind of keep it more on that, taking our measurements of these different communities and coming up with these different scales, uh, more from a qualitative thing, right? Where we're just defining or describing what we see as a more general overview. Ultimately, if we as a group decided to try to find someone in our community that would help us make some kind of piece of technology to do a scoring system like this, it's wonderful. I'm in. Ultimately, we need to have that kind of be part of this mindfulness, that we would use that as a tool to notice what's going on and to see big patterns within the agricultural space rather than get too focused on measurements, right? And for lack of a better term, sort of recipes, right? Perfect recipes. So there, I'm gonna go ahead and stop the recording, and I want us to just those of us who are in the room to sort of riff through and have a discussion about what we think in these things, and if you agree with my interpretations, if you disagree, a lot of you have more experience looking through a microscope than I do. So I'm gonna go ahead and stop the share for just a second and turn off the recording, and then we will march forward.